Good morning, students. Next topic is lipid catabolism. Under lipid catabolism, we are going to study about beta oxidation. And apart from beta oxidation, we have alpha oxidation and omega oxidation also, but it is not under purview of your syllabus. And you are going to study only the beta oxidation. And in the biochemical aspect, okay, microbial metabolism, maybe in the detailed manner, only the biochemistry aspect okay, they will study, but in the microbiological aspect, beta oxidation is more than sufficient. And before going to the beta oxidation, we can have the biochemistry of lipids, then we can go. And usually the lipids are structurally and heterogeneous level. It is a group of substances and highly non-polar, and it may be soluble in non-polar solvent only, and hydrophobic and insoluble in water. These are all the common properties of lipid you may be studying in the basic biochemistry and all. And these are all the fatty acids, because the palmitic, stearic, oleic, linoleic, and lactobacillic, tuberculostearic. Okay, these are all the different fatty acids are available. And apart from fatty acids, we have I know the glyceraldehyde, triglyceride, okay, and steroids. Okay, all substances, phospholipids, all possible, and we can see one by one. The palmitic acid is 16 number carbon, and stearic acid is 18, and oleic acid is 18, linoleic acid is 18, and lactobacillic in 19, and the tuberculostearic is 19. Next, coming to the beta oxidation of fatty acids, the many bacteria can grow on long chain fatty acids. Example, Pseudomonas, and most of the Bacillus and E. coli also. The long chain fatty acids should be hydrolyzed or further shortened, okay, to produce more monomer, okay, that is 2-carbon monomer, that is acetyl coenzyme A. So the fatty acids are oxidized to acetyl coenzyme A via the pathway called beta oxidation. And the beta oxidation, the second oxidation happens in the beta carbon. That is what is called as beta oxidation. If it is happening in alpha carbon, that is alpha oxidation. And if it is omega carbon, okay, that is omega oxidation. So we are going to study only the beta oxidation. The oxidation happens only in the beta carbon. And usually the end product is acetyl coenzyme A. And acyl-CoA synthetase complexes, okay, may be possible, okay, for the formation of different metabolites. We can have the pathway now. This is the oral pathway of beta oxidation. It's a very important question for the examination point of view. And there are 5 enzymes are available. One is acyl-CoA synthetase, fatty acyl-CoA dehydrogenase, and 3-hydroxy-acyl-CoA hydrolase, and 3-hydroxy-acyl-CoA dehydrogenase, and ketothiolase. Very simple. Okay, all the 5 reactions we can have one by one. This is the lengthy fatty acid chain. Okay, that is CH3, CH2 in numbers, CH2, CH2, and COH. Ultimately, the last one is okay, the COH, and maybe okay, this COH moiety is very important, because this is going to be reaction now, involved in the reaction now. And CH2COH moiety and interacting with coenzyme A and liberate AMP. Okay, this is incoming is ATP. ATP is converted to AMP plus PPI. And this is the energy driven mechanism. That is, ATP spends much of energy, okay, for the production of AMP plus PPI. That energy is huge amount of energy is needed to liberate the fuse coenzyme A. Now the coenzyme A is attached with the high energy. Okay, this is the high energy bond, and this high energy bond is attached with attaches coenzyme A with CO. And now this is called as fatty acyl-CoA, and this fatty acyl-CoA once formed, okay, this the enzyme is the first enzyme is acyl-CoA synthesis of acyl-CoA, fatty acyl-CoA. That's all. Second one is fatty acyl-CoA dehydrogenase. This dehydrogenase requires FAD to FADH. 2 hydrogens are removed from second, that is beta carbon, and the second carbon, third carbon, that is CH2. This 2 carbons are removed, means here 1 carbon and here 1 carbon is removed. And after the removal of 2 carbons, this will be converted into CH, and this will be converted into CH. and FAD is converted to FADH2 because after receiving two hydrogens from number 1 and number 2, these two carbons. Next, third enzyme is 3-hydroxyacyl-CoA hydrolyase. So this is for hydrolyze. That means, okay, the hydrolyze means H2O is lysed into OH and hydrogen. Okay, that is the H is attached with here. Okay, the CH attached with hydrogen, that is CH2 converted to CH2, and the hydroxyl moiety attached with the third carbon, that is CH is converted to CHOH. Fourth reaction involved in the 3-hydroxyacyl-CoA dehydrogenase, again dehydrogenase. So you have to remember very carefully the first dehydrogenase enzyme involved, that is FAD, and second dehydrogenase enzyme involves NAD. So this NAD to NADH plus H plus, and this removes two hydrogen, one is from here and another one is also from here. So this CO is formed. So one hydrogen is from here and another hydrogen is also from here, that is remaining is CO. So this is CO, and the fifth reaction is ketothiolase. Beta ketothiolase means the beta carbon involved in the degradation, that is between the beta carbon and the third carbon. So the bond will be broken down. Okay, that is completely spliced, okay, by the enzyme. Okay, that is called as ketothiolase. Thiolase means thiol bond cleaving enzyme. Okay, this bond, okay, is removed, and due to that CH3 COA CH3 COA due to valency, CH2 is converted to CH3 COA is formed, and remaining CO is attached with the coenzyme A. That is coenzyme A is going to be attached with here. That is the incoming coenzyme A. That is coenzyme A molecule going to be attached. Again, this also the energy is the breaking this bond, the liberate energy. That bond is used for the making of, okay, this bond, that is ACY. So again, now this the total number, two carbons are removed, 1 2 carbons are removed. Again, this will enter into another cyclic pathway. So each and every time two carbon is released. For example, a fatty acid, I'll show an example. The palmitic acid is having 16 numbers. The 16 numbers, okay, divided by 2, that is 8 times of 8 run of okay, this beta oxidation is required to remove all the carbon into all the 2 carbon into acetyl coenzyme A. Actually 7 times it has to go and to liberate 8 numbers of acetyl coenzyme A. So this is beta oxidation, and this is a very important topic for the examination point of view. And so this is a simple way of drawing that fatty acids, and this is not simple actually, this is a little complex also. When compared to this is better, and maybe we can omit this one. And next, coming to the anaerobic respiration. And anaerobic respiration, especially the sulfur compounds, nitrate and carbon dioxide as electron acceptors. Sulfur compound, nitrate and carbon dioxide, what is this actually? The respiration actually involved in the presence of oxygen. So this is anaerobic respiration. Without oxygen, the respiration happens. That means the microorganisms present in the sea bed or without any oxygen environment, okay, that will liberate ATP, that is energy is produced. That how the energy is produced, we are going to study about that one. Okay, that especially that organisms are highly anaerobic, and usually the enteric bacteria such as E. coli may be involved in 100% anaerobic condition, and that will utilize, instead of oxygen, nitrogen can be used, and sulfur compounds can be used, and carbon dioxide as an electron acceptor instead of oxygen. Okay, and there's a table involved in the different other than oxygen. This is the first one is oxygen, so minus 219 plus 0.07. This amount of free energy is liberated. So this is the highest one in the world. That means those animals, respirating animals using oxygen, liberates more amount of energy when compared to any other organisms. And if it is utilizing, instead of oxygen, if it is utilizing nitrate, that is minus 206, next level. And if it is Fe 3 plus 209, and CH3Cl, that is methane, that is CH4 producing one, the minus 135, and MnO2 manganese and 134, and selenium 129, and chromium 90, and As 46, SO4 20, and CO2 to CH4, that is minus 14. And these are the examples of anaerobic respiration. And so this is an example of anaerobic respiration. That is, this is the aerobic part we already studied, and this is anaerobic part. Okay, this is the um, this is organic acids. Organic acids undergo further metabolism to be formed, and it will produce either H2, that is hydrogen liberation, and also if it is involved with cytochrome B559, okay, they will be forming menaquinone. Menaquinone is the electron acceptor and produces fumaric acid, and preferred anaerobic respiratory pathway that produces nitrate. Okay, so usually this one is very famous, nitrate metabolism, and the nitrate reductase is a very important enzyme, and cytochrome B558, menaquinone, okay, all these involved in the anaerobic respirations. Next one is sulfur sulfideogenesis. So many bacteria and archaea use sulfate and elemental sulfur as their electron acceptor. All these organisms are obligate anaerobes, and sulfidogens are grouped into mesophilic gram-negative bacteria and spore-forming gram-positive bacteria, and thermophilic bacteria and hyperthermophilic archaea. That means sulfidogens are grouped into mesophilic gram-negative bacteria, spore-forming gram-positive bacteria, thermophilic bacteria, and hyperthermophilic archaea bacteria. So the component is this ATP utilizes sulfur, and this is converted into adenosine 5 phosphosulfate, that is APS. So this is a high energy molecule, and the sulfur is activated in the form of APS. So here the aerobic and denitrifying electron transport chains and paracoccus denitrificants. So the denitrificants maybe work in the electron transport chain in the presence of oxygen. Okay, these are all the byproducts, and in the absence of nitrogen, oxygen. Okay, these are all the byproducts. So nitrate, nitrite, and nitric oxide, and ultimately nitrogen. Okay, and this reaction, all the enzymes involved is nitrate reductase, that is a major contributor, okay, for the anaerobic respiration, and all the cytochrome C is anaerobic respiration based cytochrome C also. So now the end products are nitrogen, nitrate, and nitrogen dioxide, and nitrite also. Okay, and this is a model. Next, coming to the methane, that is the formation of methane, methanogenic bacterium, and the carbon dioxide is utilized, and carbon dioxide is further metabolized into methane. So we can go with simpler chart. Okay, like so here the carbon dioxide, methanobacterium, thermoautotrophicum, and by the action of the formyl, okay, methanofuran dehydrogenase, formyl methanofuran is formed, and from this N-formyl 5,6,7,8 tetrahydromethanopterin is formed by the action of formyl methanofuran formyl transferase enzyme, and this further, okay, formed N5N10 methynyl and methynyl tetrahydromethopterin. Okay, that is the substance, okay, that is formed, that is N5N10. So methynyl tetrahydromethanopterin. So this has to be spelled like that. You can separate into the different words. Methynyl till this one, methynyl, tetra means 4, hydro means including the hydrogen, and methanopterin, attraction of methanogenic. Okay, and this will form subsequently N5N10 methylene, methylene, okay, and tetrahydromethanopterin. Okay, and further, okay, this will form methyl coenzyme M, and finally methane. So this is a very complex metabolic pathway involved in the formation of methane, and this methane is the end product, and it is more flammable, and also can be used for further uses. And these are all the electron acceptors. Other than that, the humic acid, okay, can be used, chlorate, perchlorate can be, iodate, organic sulfonate, organic nitro compound. Nowadays in agriculture, okay, we are using humic acid to facilitate the organisms to utilize the humic acids to produce more amount of maybe the liberation of energy, okay, for the metabolism of the bacteria. And so with this, we can conclude anaerobic respiration. And the next one is fatty acid synthesis. And with this, we can conclude all the electron acceptors other than oxygen and other than oxygen, okay, all nitrogen, sulfur, and okay, the formation of methane, everything. Carbon dioxide as a electron acceptor. Thank you.